Okay. Hello, everyone. Thank you for joining us for the June 2025 Plant Postdocs webinar. My name is Amy Uehara, and I'm a postdoc in Dominique Bergman's lab at Stanford. If you don't know anything about Plant Postdocs is a community of postdocs that was founded by Arif and Sunil when they were postdocs. And they recognized a need to collect all these postdoc-related professional development resources and to distribute it amongst the community. Currently, this is the current Plant Postdocs leadership team, and I will be your co-host as well as Xiaohui Li, who is a postdoc at Caltech in Trevor Nolan's lab. Today, we will be talking about editing in the plant sciences, and we will be featuring Nancy Hoffman, Ellen Kuwana, and Mary Williams. The goal of today's webinar is to get a good idea of the broad editing roles that are available in the plant sciences, and to also be aware of the resources that are available to us to help us in our writing and editing. And so to begin, uh, we would I would like to ask the panel to introduce themselves in less than 3 minutes and give a quick teaser about what you will be speaking about in the webinar. So let's start with Ellen, and then we'll follow with Nancy and Mary.
Hi, everyone. Thank you so much for joining us today and for spending your time in this way. I'm so heartened that there's such an interest in writing. And of course, science is not finished until you publish your results, in my opinion. So I have an academic background. I did my postdoc at UC San Francisco, a little bit north of the people who are in Stanford. But I was always wandering around the labs asking what people were working on in terms of their writing. And I was like, oh, I'll edit that. Oh, I'll look at that. And kind of got a reputation for being good at this. So then people started coming to me, with book chapters or other things that they were writing. And so that's when I realized I really liked science writing. And Bruce Alberts at the time had left UCSF to become head of National Academy of Sciences. And he was giving as many talks as he could at UCSF and elsewhere, saying, we are training too many PhDs in the sciences. Some of you smart people need to go into science education and into science writing. And I went, oh, what is that? And so that piqued my interest in both of those two things. But I went on to working at University of Washington in Seattle for 14 years on various grant-funded projects, Seattle Children's and bioethics, but all along doing a lot of writing, including writing for neuroscience for kids, which was for, like 6th to ninth graders ideally. But we had a lot of college students using it because we didn't dumb down the text. Um, and then during the pandemic, I quit my job and went freelance. And have not looked back. So I'll tell you a little bit more during my slides of what type of work that I do. But that's me. And I also have an interest in AI assisted tools for helping you edit things safely. So I'm going to be talking about that in my talk.
Great. And thanks everyone for being here. I'm excited to join you all. I also have a background in plant biology. My PhD was at UC Davis. And again, similarly, was always helping people with writing, doing writing. I ended up after staying home, having kids, ended up working as a science editor for the Plant Cell, meeting some other science editors there, and then deciding that we actually really enjoyed the editing and the freedom of editing. And so we started a company, Peridot Scientific Communications, but it's also known as Plant Editors, that now is a team of about 10 to 15 editors. We think of ourselves as a boutique editing service. We focus specifically on plant biology, mostly cellular and molecular. We do a lot of pre-submission editing for individual authors, and then we also do post-acceptance editing. And that's what I'll talk about more today. And then my last little bit when we go into the exercises part will be focusing on like my big thing to writer or to authors is to make it easy on your readers. So do the work for them. And I'll talk about that a little bit more.
Hi, I'm Mary Williams, and let's see, I guess my journey began with doing a postdoc at Berkeley, not far from Stanford. And then I went to Harvey Mudd College, which is just down the road from Caltech. And it's a small liberal arts college of science and engineering. And I was a professor there for 14 years. And what I really learned there was how to teach people how to write, because we had a very writing intensive science curriculum. So from their first year, the students were writing. And so I didn't really know how to teach people how to write, but I quickly learned because that was a big part of my job. And I found that I loved it, and that it is so rewarding to help somebody become a better writer, because you're really giving them this life skill that's going to help them no matter where they go in their career. So then in 2008, I came to Glasgow, Scotland on sabbatical, and I decided never to leave. So suddenly I couldn't be a professor in California at the same time as living in Scotland. So I got offered a position with the American Society of Plant Biologists as features editor for the plant cell. So when we were discussing this, editor means so many different things. So I kind of have two things to talk about in the presentation today. I'm going to mostly talk about what an editorial board does, which is different than what Nancy and Ellen are talking about in their roles as editors. But I also have another job that's not that dissimilar from what they do, which is I work with the assistant features editor. So my title is features editor, but we have a whole group. We have 34 assistant features editors, mostly postdocs, who write summaries of the research articles coming out in our journals, Plant Cell and Plant Physiology. And so there I'm doing the same thing. I'm helping them become better writers. I have to say that the vast majority of our assistant features editors, English isn't their first language. So, they are a little nervous, and we're like, no, it's the ideas. We can help you with the grammar. Don't worry, get the ideas straight. And I'm sure we'll hear more about that as well. So I guess that's all I have to say, but I'm looking forward to the presentations, and I think it'll be quite enjoyable.
Yeah, thank you. Thank you for all the panelists to introduce yourself and give us a hint about what you're going to talk about in the next hour. So we're going to start with our first section of this workshop to get to know more about the editing from the beginning to the end. So we have some scheduled questions that we're going to start with. So first one is, so where in the pathway from paper draft publication do you find yourself, and how does your job relate to the others as an editor? So for this part, we'll start with Mary.
Okay, so as a member of the editorial board, I'm involved in helping to select the papers that will be published in the journal. And so I'm going to really talk about how that process works. So this is after all the good work that gets done to polish the paper, make it beautiful, and submit it. So this is the way the ASPB journals work. Different journals work differently. But what happens is when someone submits an article to the journal, the first thing that happens is an editor receives it. And the question we ask ourselves is, does this paper seem like it might be published? Because as you know, people are overburdened with peer review. It's a lot of work, and people don't get paid to do it. It's part of their professional responsibilities. So we don't want to send something out for peer review if it doesn't look like it might get accepted into the journal. So we have a big editorial board on both of our journals, and those editors are full-time scientists. So they are also working for free. They get a tiny honorarium, but it's pennies per hour, really. But they do it because they're committed to the science. So we pull in the appropriate editors who are maybe working in the same field, and we just say, have a look. Do you think this looks like a good fit? So we call that a pre-review consultation. And then we have a little discussion. It just all happens in a little online form. And let's see, I should be able to move the slides, but I'm not. Oh, there we go. It's just a lag, I think. So at that point, a decision is made, and we try to do that very quickly. Either we say, yes, we're going to send this out for review, in which case we go to the next step, but sometimes we don't. And that happens maybe half the time. And if the paper is not a good fit, if people are worried about the conclusions, if it looks like it's overinterpreted, if it just doesn't fit the scope of the journal, we'll try and get back to the author right away and say, no, this isn't for us. Find somewhere else. Because what authors don't want is they don't want their paper stuck in this sort of like space where they can't submit it somewhere else, but they don't know what's happening. So we try to do things as quickly as we can. Let's see if I can. Oh, okay. So if the answer is yes, we will send it out to reviewers. And again, these are people who are ordinary working scientists. They're experts in the field, but they're doing this out of their own goodwill. And that's why we don't want to send them stuff if it really doesn't have a chance. So what the reviewers do is they read the paper and they make comments on it. And they may say, you know, these are the strengths, these are the weaknesses, this data isn't presented well, the methods aren't appropriate. I'd like to see a thorough discussion of this. They're using the wrong nomenclature. There's a lot of things that reviewers pick up on. And one of the things I really encourage you to do is to find some of these peer review reports that many journals publish and just kind of read through what it looks like. Because if you're not in the position of having had a chance to experience these, they can be a little startling and a little scary. Like, oh my God, they hate it. No, they don't. They're trying to help you make it better. So the 3 reviewers write their comments, send them in to us. And then we have another consultation. And in this case, the editors, the reviewers, they don't know who they are. Okay, so it's reviewer 1, reviewer 2, reviewer 3. What they get to do is they get to read each other's reviews. And sometimes you have a situation where one reviewer is like, this is great, I love it. And another reviewer says, this is garbage, I hate it, right? And then the reviewer in the middle says, well, I like this part and this part, but not this part and this part. So this is the really hard part for the editor, because the editor is the person who makes the decision. The reviewers advise the editor. So the editor then looks at this and says, they have to really think, like, where's the happy medium? Where does this belong? What is important? What is the most important? Does this one have a grudge against somebody, and this one maybe is their best friend, which of course they shouldn't be reviewing each other's papers. But so they have to get the conversation going, like, reviewer 1, you said this method was inappropriate. Reviewer 3, you said this was the right method. Can we have a conversation so I know what to do here? And that's actually really, really useful, because in the old days, before these reviewer consultations, the author would just get the 3 different reviews with no synthesis. So they'd be like, I don't know what to do. This one loved it, this one hated it, right? So the editor actually picks the parts that they feel are critical and says, I want you to do A, B, C, and D, but don't worry about E and F, right? And so the author gets that information, and that guides them as then they resubmit their paper. I think, do I have more slides? Oh yeah, okay, so then the editor writes back to the author. They either say, you know, please revise it, which is very common. They might occasionally say, we accept it with minor revisions, but that doesn't happen too often, because it does mean it's in. You just have to fix a few things. Usually we'll say, maybe, possibly, could you fix these things and send it back, and we'll have another look. That's the most common outcome. And then sometimes we just say, you know what, we thought it might be a good one, but the reviewers really just they pointed out things that we didn't see. Sorry, find another journal. So that's kind of how an editorial board works in that version of editor. So there's 2 quick things I want to point out before I turn it over to the other folks. I mentioned that we have assistant features editors, and this is such a fantastic program, because what we do is when a paper gets accepted, we share it with our panel of assistant features editors, and we say, do any of you want to write for plant physiology, it's called a news and views, and for plant cell, it's called an in brief. We say, would you like to highlight this paper by writing it? So that somebody will pick it up, say, yes, I like this, I want to do it. And then we have a panel of editors who are assistant features editors, and they then help the person polish their writing. So the assistant features editors may get 3 or 4 or 5 articles in the journals every year just by highlighting these things. Some journals have professional editors do it, but we're society journals, so we encourage the society to grow, you know, and develop professional skills for our early career scientists by hosting this program. So currently we have an application open for the assistant features editors for plant cell. The QR code will take you to the web page, and it's a fantastic program. I really encourage you to do it. And then the other tip, we were supposed to have a tip. This is my favorite book about scientific writing. If I wrote a book about scientific writing, I would write this book, but I don't have to. I just tell people to buy it. And it's like $20. It's great. And it has exercises and samples so that you can really actually interact with it and learn. So this is my tip, is if you want to become a better writer, buy this book and go through it, and it will help you hugely. So thank you.
Thank you, Mary. We'll move on to Nancy to talk more about post-editing process.
Okay, I just requested remote
Yeah,
control.
give me one. Okay.
Okay, sounds like
Good
it.
work.
Okay, great.
Okay. Oh, sorry.
Okay. Yeah, so I will talk about post-acceptance science editing. Like I said, we spend a lot of time doing pre-submission as well, but for this part, we'll talk about that. So after everybody in the editorial board has done their bit and the reviewers have given their reviews and the editor decided to accept the paper, this science editing step happens, and it does not happen everywhere. So not all journals do this step. It basically slots in between peer review and production, like the copy editing, making the proofs, that's after us. This science editing step is one that generally, in our experience, has been done by society journals or other journals that are willing to invest in curating the literature. So what we do at that stage is we'll edit the text for I'll talk a little bit more about what exactly we look for, but for presentation, adherence to author guidelines, things that the editorial board thinks are important and might get missed during peer review. So really to standardize what ends up in the final product. We also do things like checking that people didn't just rename a gene because they wanted to rename it, but there was a good reason to do that. So really curating, keeping like things from proliferating in the literature that could be a problem. We also check figures for presentation, and I'll talk about this a little bit more, but more and more we're doing checking for best practices in the image handling. So we're spending a lot of time on figures these days. Then after we do all those things, we return those files to the corresponding author who will review the files, respond to any queries we have, and then upload revised final files that then can be sent to production and copy editing. Oops, let's see if I can make it move. Okay, there. So the kinds of things we would look for during that text edit would be, we focus a lot of the time on that first page. So it's not a deep pre-submission edit, but it is trying to find the high value targets for making the title good. It needs to, you know, if there are numbers of like word limits or character limits, we need to work with those and then make sure that the titles are the way the editorial board wants them. So in this case, I say no species name. That's something that's changed over time. Sometimes people say like every title has to have the species name. Other times that's changed and they said we don't want them in any. So that stuff varies by the priorities of the journal and of the editorial board. We look at the running title, the summaries, the abstract. We really make sure those are complete. Like, so in that case, it definitely should say what the organisms are studied, what methods are used, what approach, make sure the broad question is framed. We might do a light edit for readability in the main text, also checking certain nomenclature things, making sure things are clear. One thing we do look at in this is like the figure callouts, and not just like that they're in order, or that if it refers to panel 1, there is, or 1A, there is a panel 1A, but more if it says it's looking at protein, is that what's actually shown in the figure? So those are like we try to do the things that copy editors are going to struggle with. So, and those callouts often change during rounds of revision. So I mean, a copy editor can say, yes, there's a panel 1A and it was called out in order, but we might be able to say, you know what, that doesn't look like it's the right panel, like it's not the right kind of science shown there. And that's going to be pretty hard for them to catch. But those are the things that often do get messed up during rounds of revision. So we do that, any other kinds of footnotes and things like that. And then another place would be making sure methods are complete. In terms of replicates described, primers included, accession numbers, just all that kind of stuff that, again, may slip through peer review. In the figures, we try to help those be really well readable. So one of the big things we do is the color scheme consistent? So does blue mean the same thing throughout a figure, and preferably throughout the paper? Because it just makes it so much easier for readers to figure out what's going on if the same thing means the same thing throughout the whole thing. And that, especially with papers that are put together by different groups, sometimes you can really tell that one group made this figure and a different group made that figure. So we do a lot to help them be polished, make sure they all use the same fonts. Are they consistent? Make sure that everything's big enough to be seen and the labels are clear. We also look at the legends, make sure everything's defined, error bars are defined, it's like adequately described, there's not too much interpretation, just things like that that are the preference of the journal. And then, like I said, in these times, it's been interesting, but we've shifted for some of these journals almost completely to just image analysis. So basically, we're assessing images in all figures of the paper for best practices. So how is there splicing that's not been labeled? Is background shown so you can tell where the borders of the image are? If you've like pulled like say a leaf or something off of a background and just placed it onto a black background by extraction, have you disclosed that? Similarly, if you've made a composite image, have you disclosed that? The idea being that readers need to know whether this was one picture. So even if you're talking about leaf size, but somebody else wants to know, like, were they different shades of green? Can they tell that because it was one picture taken at the same time, or was it a composite of a bunch of different pictures? So sort of making sure that those things are clear. And then duplications, manipulation, things like that. So things that could be like problematic scientifically versus also just kind of educational scientifically. And yes, also like red, green and stuff like that. So we give people to resources for that if we need to. Yeah, so that's where I'm done with the post-acceptance stuff. I will give it back, control back to you.
Yeah, thank you, Nancy, for
Mhm.
telling us more about the post-editing process. So Alan works mostly on the pre-submission editing process. We'll give the stage to Alan. I will stop sharing here.
Okay, and hopefully you can see my slide set. Sorry, I was the person working too much this week and didn't have time to get my slides ahead of time. But yep, I'd like to start with a land acknowledgement. You're
I'm
right.
not seeing your slide.
You're
Oh,
not
okay.
seeing the
Oh,
slides?
no, I see. Okay, I see now. Thank you.
Okay. I'm in Seattle on the traditional lands of the Coast Salish peoples, past and present. So again, thank you for having me here. You'll get my slide set. I have a lot of slides. I want to give a lot of information. I'm a teacher at heart. So you'll get these, so don't worry about taking notes or anything. I edit for several journals, and I put in some Stanford connections. I was Mark Teslevine's first lab technician. I sequenced the netrons at UCSF. And then in the neuroscience department, if you know Brian Knutson, who's a wonderful neuroscientist, I went to undergrad with him, and then I shared my socials. As I said, I'm a teacher at heart, so there is a difference between Gen AI and AI assisted tools. I'm going to talk more about AI assisted tools that you can use very safely for editing before you submit your journal article, and some literature search tools that hopefully you know about, but you might not. And I always like to ground my talks in the thought that if you are bilingual or multilingual, it takes you a lot longer, or twice as long, to read and understand scientific papers. And then when non-native English speakers, writers are writing those papers, it takes them more time to write them. They're more likely to get rejected, and almost 13 times more likely to be requests for revisions on those. So I think some of these AI assisted tools can kind of level out the playing field in terms of helping to people to get information quickly, help with the synthesis of that and how that information is usually presented. Of course, lots of caveats with that. Know what you're using, know if it's uptaking your text. The tools that I'm going to talk about right now do not save your text. They're not connected in any way to large language models or other size models. One thing that I think people don't use enough is within Microsoft Word, there are editor functions, and we go and we check the tools, and we do maybe spelling and grammar check. But if you go beyond that and scroll all the way down to insights after you run those grammar checks, there's something called the Flesch-Kincaid reading analysis. And the bottom three numbers are really what you want to pay attention to. And I apologize, this is a bad example. I ran this like on a resource sheet of like a book list, so it wasn't complete sentences. But it gives you this idea of ease, and you want that number to be greater than 60%. So think of like a typical grade, in college. And then the grade level is, of course, this is between 8th and 9th grade, and then percent passive sentences. And you can run it just on a paragraph, and like, of course, if you spell out DNA or HIV or something, the reading level is going to go way up. So you can make adjustments and just run it on the text that you're kind of revising and working on getting the reading level down. I think we've probably all heard of Grammarly. There are different tiers within that tool, and there are some even that are HIPAA compliant. But the free version is actually pretty dang good. I would say, 95% of the time, I'm like, oh, okay, maybe that's a good change. I find it annoying, so I don't use it a lot, but it would be good to run the free one, on something before you submit it. They were also adding Gen AI components to it. So I try not to use those because of the energy usage. And again, you'll get the slides. I don't want to go over the privacy, but this other one, sometimes we have a lot of subheads or you're not sure what to capitalize in your title. Just run it through Capitalize My Title. You can toggle between different style settings, style guidelines, and then it also has all these blog posts and tools for writing and editing specifically that you can play with. And then if you really want to get more help with your writing, there is a tool called Perfectit that runs on AI-assisted algorithms. It is not uptaking your information, so you can put confidential things in there, things that haven't been published. You need to log in with the same password and email that you use for the cloud from Microsoft Office. It embeds in the ribbon, and it checks hundreds of things within your text for consistency. It's smart enough to know you might have abbreviations defined twice, both in the abstract and the body of the text. And it puts you in complete control. You can fix things with one click of a button, like all the numbers to numerals, or you can go and look at each individual case. I'm also going to show you Edit GPT. Again, you'll get these slides. I'm going to go through them pretty quickly because I don't know what's relevant to which people, but Edit GPT is not related to Chat GPT at all. It's a standalone created by one person, does not take up your text, very safe to use, has a free and a paid version. And then if you're struggling with writing, there's an academic editor in the UK who has created a tool called Really Write that works kind of like another tool called Hemingway that color codes your text. So it puts all the adjectives in one colors and all the adverbs in one colors. Those are those extra words that if you're fighting for space, you might be able to remove. You don't have to say we carefully dissected. You could just say we dissected. And then it'll put complex sentences in orange or red. Those are things you want to take a second look at. See if you can break them into 2 sentences. So this is Perfectit. Again, it just sits within your Word window. You run it. It works right in Word. So you're not going to lose text. It flags these inconsistencies. It's not a spell checker per se, but it will check, it'll find if you have manager and manger in the same manuscript. It'll find that and flag that as an inconsistency. Same if things are italicized, not hyphenated or not. And again, it's smart enough to know you might have something hyphenated like T-cell therapy, and then you might say this affects T cells, and you might not have that hyphenated. So it's smart enough to know that. These are all the things it checks. Again, I don't want to belabor that point. And then I think I have a quick demo here that just shows you how quickly this works. And you can imagine if you have multiple authors and people are using different terminology, if you have a British author, it'll spot those spellings. It has lots of style guidelines already entered in it. So if you're writing something for the World Health Organization or something like that, you can go right to that. Or if you're submitting to Nature and you want those, British spellings in there, you can toggle to those different style guidelines. And again, it's just calling out these different things, and then you can click if you want to go and fix it or go to next. And then at the end, you can do all these things such as double spaces to single spaces always. And in hundreds and hundreds of pages, it will implement that one change. The other thing that I like is it will give you a table of abbreviations. So when I'm working on a grant or a manuscript and I'm editing it, I'll copy that and then go up above and put that in a comment so everybody knows what abbreviations have already been defined and they can just jump in and start writing. So that's Perfectit. You can do a free trial, I think, for a week or 2, and then it's a paid subscription. Edit GPT is a tool that I'm really, really excited about. Again, created by one person. Your text stays private. You can put up to 10,000 words in a month for free. And it can do all these different things. And then starting from the upper left and moving on, the level of edit gets deeper. And we've talked about this a little bit, that editing means a lot of different things to different people. And like when somebody asks me to edit, I think they think I'm looking at only commas and not the words. And like, no, just like make sure the commas are right. And I'm reading, thinking developmentally, what's missing, what's wrong, what's in the right place. And then line editing is each sentence doing its best to present the information. And then like paragraph level, should this be broken up into two paragraphs? Does this make sense? So there's a lot of things going on in editors' heads. This will start to look like Christmas here, a lot of red and green. You can accept each change individually or all at once. And as you go deeper along the upper left-hand side, you will see that it's making more changes. Some of these you will not agree with. Like at the very bottom, I put in the symbol for greater than 200 institutions. I would not change that to over in scientific text and abstract when you're really fighting for space. But involves the use of to uses, that's a great change, right? Things like that, such as and like, have two different meanings. So again, I wouldn't change it to like. Like is you're comparing it, such as is including that example in the group that you're talking about. So again, you would need to go through this and it takes a little bit of time, but this is like having a writing instructor or a good editor. And my hope is that if somebody feels like they're not good at writing, this would bring it up to a level like when you hand it into your PI, they can really do that more substantive developmental edit. And what I was really surprised by when I put in an R01 grant proposal, that was honestly the most difficult grant I've ever worked on. It did a really good job making it more concise. And I put it in after I'd edited it just to see what it would suggest and see if I'd missed anything because you can't catch everything, especially after you've read it a lot. So what I also like to do is combine it with the Flesch-Kincaid reading statistics. So here's the starting text. And as you can see, I have to move you all out of the way. This started at 37 words per sentence, which is really, really long. The easiest way to lower the reading level is to break your long sentences into two sentences. And then our reading level is really, really low. We're like at 2, and we're at 22nd grade. So we're 6 years out of college. And then from there, I asked EditGPT to run it through the concise functionality. Now we're down to about 30 words per sentence, which is better. We've improved the reading ease by a factor of 4. We've gone from 2 to almost 8, and we're at 19th grade level. So we're in grad school at least. But a human editor can still do a better job than these tools. Thankfully, but it gets us a long way there. So you can see we're at 22 words per sentence, which is a huge improvement. We are 7 times better than the original text, and we're at a 16th grade level. So, 5th year of college. So that's really pretty good. And again, you'll get these slides so you can kind of see the changes that were made to make things more concise and easy to use. And then all I want to show you here is how quickly this program works and not to panic when it turns everything red. That's its way of showing you what it's working on. So there we are. We're done. That's all of it. And I forgot to scroll up because this was so big. Oops. Okay. So that's the demo. You can see it's just very, very, very fast. Literature search tools, if you haven't played with these at all, most of us just go to PubMed, and I'm almost done here. But there's one called Cite AI that was built with NSF and NIH funding. So it's one that I really like. It has, instead of going and searching like ChatGPT and searching everything out there, you're searching a skyscraper library that has 2 million papers in it and 1.3 billion summary statements. What I like about this tool, it's tracking corrections and retractions. And when it cites that summary and you click on the superscript number and it takes you directly to the paper, it's giving you the text around those statements. So you can see if the statement is agreeing with your query or contrasting it or is just neutral. And again, this is going to look very familiar to anybody who uses PubMed. You can refine your searches in any way. All these tools are adding Gen AI components. Just know that ups the energy usage, 8 ounces of water for each prompt that you put into Gen AI. But you can ask more like association, correlation, how do things relate to each other. You can save searches in different folders, and you can also create custom dashboards so that it's always searching within those 2 million papers and pushing information to you as more papers are added based on your areas of interest. Another tool that's completely free and then also has a paid version is called Consensus. It's very similar. It doesn't look quite as slick, but similar number of papers. And then I always like to see who's involved. To the left of the blue-haired person, Jevin West is a professor of mis and disinformation, as is Dr. Christakis on the upper left. So they're really trying to put good information in these tools. These are example prompts that you can use. This is how it works. This is what it looks like. Again, it looks a little bit more, it doesn't look as slick, but it works just as well. And you can toggle all these different things, and then you can ask pretty intensive questions. So I think maybe I'll stop there, and then I might come back to some Gen AI. I don't know why these are, oh, it's just, I have it on a click. As I said, it's a high energy use for these Gen AI tools. So I think I'll stop there because that's a lot of talking, but you'll get these slides.
Yeah, thank you. That was a very nice overview of like the editorial process and all of the different roles of the editors from everyone and the tools available. I think we want to capitalize on the amount of experience of editing that we have in this room. And so we would like to get your feedback on like what skills are required to be an editor with the perspective that maybe some of us may want to go into scientific editing in the future. And so maybe we can start with Nancy, Mary, and then Ellen.
Okay. I would say for my kind of editing, rather than sort of Mary's, one of the big things, I mean, obviously we need knowledge and we need attention to detail and, somebody who can really think about how you're communicating and awareness of audience, all the skills. But the other piece you need is to be able to set your own ego aside and not be the author, right? So we're working on someone else's message. We're honoring what they want to say, and we're helping them get their message out. We might help them refine their thinking about what they're doing and push them to think about other things. But we're acting as a proxy for the reader when we ask that, not in terms of what they will need to know or what they might be wondering. And the authors can not go the direction we think they should. And that part is a skill. It's like you have to accept your role, like where you're engaged and passionate about it, and also it's not yours. That's true for editors in all different fields, but that it's a really different skill than just writing, and it's a different level of ownership, I would say.
So I'll talk about my role in working with the assistant features editors. And what I do and what the other assistant features editors do is we don't read the manuscript. We start with their summary, and we read their summary and we say, is this interesting? Have they captured my attention? Do I understand what they did? Do I get bored halfway through? And that's never a good sign when you're trying to read something that's supposed to be promoting some exciting science and you find yourself drifting off and looking at, Instagram or something. And so being able to put yourself in the space, is this interesting? Did I learn something from it? But the other part, because we're doing this as a professional development for the assistant features editors, is you have to be able to give good constructive feedback, right? You don't just change it, you're teaching them. So again, I think for my role, there's a lot of teaching involved. So, I think you should consider breaking this up into 2 paragraphs because, or people really read the first sentence of a paragraph more carefully than anything else. You should put this key point at the beginning and then fill in the details, that kind of stuff. Yeah, and I think for being an editor on an editorial board, sometimes egos do become problematic. And I think that, having an editorial board where the people are generally nice and supportive of early career people, because most first authors are early career people, right? And when they get nasty comments back or harsh treatment, that's not good. So I think, again, being kind of community minded for a lot of the work we do is very, very valuable.
Yeah, I love this conversation. I will say, yeah, try to look at the editing process as a collaborative one. I come in after the scientific reviewers have accepted or given a provisional acceptance to the manuscript. I work with a lot of multi-bilingual authors. I try to preserve their voice. I don't want everything to be homogenized to ChatGPT voice. And also know that some of the people editing your work, might not have a super strong scientific background or might inadvertently change something. So after editing, you really do have the responsibility to take a careful read and make sure things have not been changed from your meaning. And what that means, if somebody changed your sentence, it means it wasn't clear to them. And so that's a flag for you to go back and try to make it more clear. And when I do my edits, if I'm not clear on what the sentence is saying, I will put a comment and I will say, if you mean this, use the comma here. If you mean it this way, do that. And I think, everybody's so smart. Only 3% of the people in the US have a PhD. There's no reason for any of you to have imposter syndrome. You are incredibly well trained. You are not necessarily trained in how to convey your science clearly, and so much of that is dependent on your PI and the senior postdocs as you were a grad student, and that's not a standardized training environment, right? So you might have had a really good scientist who was super busy and didn't give you a lot of feedback on your papers, and you have to assume they're fairly good, but that just could be a limitation of that PI's time. So really try to look at it as a collaborative process. Don't take it personally, but you are also the advocate for your text, and you may need to go back and say, no, I meant this, and I want to phrase it this way. Does this make sense to you? So there, you have to build in some time for that back and forth. But I think the editors can really help with, as again, a stand-in for the reader. Is there enough context? Is there too much jargon? Are things explained? I see a lot of just like lists of things. And then I'm like, oh, these are key transcription factors. And e.g., we're going to put in 3. We're not going to list, 20 of them. And then use your transition words to help guide the reader. Think back to when you were a beginning grad student and you loved reading review papers because they were so clear. They summarized things at the end. They gave you the main point. They kind of walked you through and told you this is the most important thing. And then also think about in journalism, they do a very good job of inverted triangle, which is putting the most important information up first. We know people read in an F shape, as in Frank. I need to think of a plant name that starts with an F. So you really want to front load not only your page, but the beginning of your paragraphs and the beginning of your sentences. We start with these long phrases and caveats and limitations, and then 12 words in, you have the subject. Start with the subject. Even saying there is blah, blah, blah. There are 52 cards in a deck of playing cards. You can just say a deck has 52 cards. You know immediately what we're talking about. Grab readers' attention. You can say, surprisingly, we found. You don't have to do all passive. So really try to think about kind of like a narrative voice and how you would explain it to somebody who's not a scientist and weave some of that in. Okay, I'm going to stop talking there, but I'll put some resources in the chat too.
Yes, thank you. Next we want to talk about, let me see, the slide is not moving. So next we want to talk about what strategies do you have for scanning documents when initially starting the editing process? So we can have 1 or 2 minutes for each panelist to talk briefly about this process.
I can start again. Especially when I started editing, I think I needed to get a broader sense of the whole paper before I just dove in. So at that point, I would, look at the title, look at the section headings, read the abstract, and move through it that way. Look at the figures maybe to have a sense of where we're going with this. Since I've gotten more experienced, I haven't needed to do that so much. Have a better sense of where things are likely to be going. And so I can just kind of dive in, look at the title, and then dive into the abstract. If the abstract seems like kind of a mess and I can't tell what their argument is, I might go to the introduction and try to go there. And if it's all really not coming clear to me, then I would go look at the different sections of the paper first, and then come back to the introduction and then do the abstract last, once I'm really clear on what they're doing. The other place that sometimes people have a hard time writing an introduction, they get it turns into a review and it's not focused enough, but lots of times people write a really nice summary of what they did and why as their first paragraph of the discussion, where it's like I don't think that in general, I usually tell them they don't need to reiterate everything, but often that summary that people have done is much better, and we recommend moving it to the front. But that's another place that sometimes if you're having a hard time telling what the point is, looking at that paragraph there or the end of the introduction, these places where people give their message are places that I would start if I'm struggling to find the point.
Do Mary or Ellen have anything to add?
Sure, I can go next. I heard one editor who runs the document through Perfect It first, just to see how many things it flags, and that helps them to kind of have an estimate of how much work the document might need. I think you can kind of do that visually as well. But as Nancy said, I have a slide that says, it just has 3 words. It says four, sit on your hands. And literally, I want to line edit. I want to go in there and fix everything as I read it. On first read, I really do try to put my pen down, sit on my hands, take my hands off the keyboard, and just read it quickly for that macro overview, because I tend to go really micro, and that's just the way my brain works. Quick look at text and figures. Oftentimes, people they should do their figures first, but they really spend most of the time writing, and then there's too much redundancy. They've really described in text what's represented in a table, and so trying to help them pull that back to a more like, what are your main points, what are the trends, and then people can look for the nitty-gritty of all those details in the table. Reverse outlining is something when you start writing and then you get kind of lost. And as Nancy said, maybe something from the discussion should go up in the intro. Reverse outlining, you can either kind of print it out and on the left margin, look at each paragraph and write, what is your topic sentence? What is this paragraph about? And then on the right-hand side, read through that paragraph and bullet point out your main points. This talks about this, this, and this. And, really you want no more than 3 bullet points, right? Are those in the right order, priority-wise? And should something belong in another paragraph? That's a really fast way to build an outline after you've already written it, because we all know we stray from our outlines.
And
So I think those are my top tips. And then if you put it into like ChatGPT, Claude AI is a better writing tool than ChatGPT. And then think about which section should be going from broad to specific, like your intro. And then in your conclusion, you're going from your specific results to a more broad statement, and working with kind of those no words instead of maybe emphasizing that this might help an underserved community. You do that broad statement of this will improve the health of, every individual in the US, something similar to that.
yeah, thank you, Ellen. And then Mary, if you have like something to add in about a minute.
One thing,
Yeah.
I love a summary figure. So I always look at the very back and I see, did they make a summary? Because if they did, then I can kind of like follow the text and say, oh yeah, that's that endoplasmic reticulum contact point there, right? So I love those. Make more of those.
Yeah, I love those too. And I think we have a little bit of time. We're trying to respect everyone's time and finish around 11. I think Nancy prepared some practical tips and tricks for us. So if we wanted to just jump into that, and hopefully we'll have some time for questions at the end.
Oh, okay. I have remote control, looks like. Okay, I'll try to go through these quickly, but they had asked to have some exercises. So I just wanted to point out some things. The main advice, as I said before, that I have for authors is that you need to do the work. You're asking your readers to pay attention to your thoughts. So make sure you have done the work and make it as easy as possible. Make your message unavoidable. One way to do that is, can you write your cover letter before you start writing? Like, what's the point? What are you going to say in 2 sentences, 1 or 2, about why this work is important? And then make it super easy for readers to find that point in the paper. So places that you can do that in your manuscript. The title is obviously the first one you can do that. So I was going to just go into titles a little bit. So you could write a, boring effects of this on that kind of title, or you could write one that says the answer. Arabidopsis roots required a threshold level of something to do this. And I'll come back to these a little bit, but The other thing I would say is like, you can also have a title that just reports the mutant shows this, but is that what we really care about here? What we want, I really encourage people to, unless there's some reason the mutant itself is important, to turn it around to say, what did we learn from this? So this factor promotes stem growth. So what do we know about the wild type from this, rather than just sort of reporting the result? I'm going to come back to that a little bit more. So like almost 10 years ago, the editor-in-chief of Genetics wrote his thoughts about how to write titles. They're pretty specific, and I don't always use this advice, but I do think it's something useful to think about. So his argument was that you want the interesting topic first in the title. So it's not just, like it says, a bunch of results. So his thing is start with the topic of the paper, not the name of the thing you studied. Make it exciting, like express in the title why you think this is worth publishing, what's new about it or what's interesting. And you don't have to overstate it, just accurately state it, but make sure you haven't left it out. Sometimes I edit a paper and then I look at the cover letter and I was like, oh, that was why this is a plant cell paper versus a PLOS paper, whatever, like, so, but that wasn't in the paper. So you want to make sure you include what's exciting about it. Avoid too many abbreviations, keep it short. And then his advice is to not give away the ending. I don't always think that that's necessary, because sometimes I guess you have to decide what's your goal with the title. If the title is to get someone to read your paper, then his approach is good, where you just say there are multiple barriers to something and you have to read the paper to find those barriers. If your goal is to make sure your paper gets cited for this fact, you might want to put that fact in the title, assuming that someone may not read any more than the title, which would be not great best practice, but at least even if they've read it later and they forget why, if you want to just say, this is the thing that I want everyone to know from this paper, you might put the whole story in the title. So let's see if I can get it to move. Okay, let's see. Sorry, it's moving really slowly now. Okay, there. So, okay, if you go back to those titles from the last one where we switched it to saying a message, if you did his approach, you might, rather than saying it needs a threshold level, you might start with maximal root growth as the thing that somebody might be most interested in, and then say that requires a threshold level of something. And similarly for the other one where we switched it around to say what happens, we might say something like start with stem growth, and then say like, why is it interesting that that's needed? It's a non-canonical translation factor. So something that adds a little interest. Why is this worth reading? Those I think are more enticing titles. The other thing I would say is that make it easy for readers to understand your point. So when you give them information, say what you conclude from that information and say why. One sort of place to do that is if you find yourself writing consistently, like give one piece of information and consistently another piece of information, rather than consistently like it happened all the time I checked it. That's a place for you to increase the connecting logic. So lots of people change that to just consistent with this, which is like, okay, but it would be better to say like why, consistent with the role for X and Y, stems were longer when this happened or something. So it's a chance to just sneak your logic in there. Doesn't take a whole lot more words, but it sometimes does take more thinking, but it really helps your reader to know why are those two facts connected, because sometimes you read papers and it's really hard to tell that. Other places to state your conclusions would be in the abstract. So rather than saying like at the very end, I'm going to discuss a model, say like, what's the summary of that model? Why promotes, just say like, our results indicate this big thing about the model. The end of the introduction, that last paragraph of the introduction shouldn't be another abstract. It should clarify the questions you addressed, the hypothesis you addressed here, the broad approach you used, and we recommend that you put in your conclusion as kind of like foreshadowing the rest of the paper. So what you want readers to remember about this work. Not everybody agrees with that part, but I think it really helps readers to have a sense of where you're going before they read the rest of it. Also section headings in the results and the discussion, if you have section headings there, which I really like as a reader, is the same deal. If you just say we did expression analysis of these genes versus saying this one is expressed in stems under salt stress, like, okay, it's clear you did expression analysis to get to this point. You probably looked at more settings. Like, so it just gives so much more information and helps pull out the piece that you want readers to keep track of. Similarly, figure titles can state the conclusion. The experiment can be described in the legend, but that you can use that title to state what you conclude. And these, I think, are places you can check the logic of your paper. So if you look at the section headings as a group, do they lead to the conclusions that you're wanting readers to get? And then the last thing, because I'm trying to be quick, is focus at the paragraph level within paragraphs, focus your text. So when you first write, or when I do, I tend to just write everything. But then when you're at the editing stage, if you're thinking, this is just a link to a blog that we have about how you decide what to include in your introduction. But the reason I bring it up is, so I put in all these details about the experiments someone else did before, which is useful for you to keep in mind. But when you're looking at your introduction, are these things important? Do we need to know how they did it? It only matters is if your experiments go to, oh, we got two different, you do it two different ways, you get two different answers, then yes, the method might matter. But if the answer is just they interact, then that's all you need to say in the introduction. And so we just have some questions that we keep in mind of like, do these things address these questions? And if so, they belong in the introduction, and have they been fully addressed? And then the last thing is when you're focusing your language. So here's one of those like terrible sentences, figure 1 shows protein accumulation, like blah, 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 blah. You want to tighten them, but the way you'll tighten them is different in different sections. So in the results, you might tighten that to after 5 minutes, more of this protein was in the mutant than in the wild type, and refer to figure 1. In the discussion, you might go further to like, what do you conclude from that information? That X, the wild type X, represses that accumulation early in the drought response. So what does 5 minutes mean? You want to tell the readers, why is 5 minutes, does 5 minutes matter? Why does the mutant matter? So those are the kinds of things I would think about during that last step of going through your text. And I think that was all I had. Yeah, I'm done. I'll pass it back over.
Thank you, Nancy. I think Alan has 1 minute of interactive session to share, right, Alan?
Sure. If people are willing to put up with that, and then you can think about any questions you have. I was just gonna say again, if you can get your information from a simple internet search, it uses 10 to 30 times less energy than generative AI, which can hallucinate. Something somebody put on the internet 7 years ago about Kylo Ren syndrome as a joke came up recently in a search they did. And it's hard to tell fake information from real information. Recently, last month, if you put in an idiom and asked ChatGPT to explain it, it would just do it with the confidence of a, 16-year-old, just making things up and fooling their parents. So I'm not even in presentation mode, but that's okay. In your prompts, do not say please and thank you. Use the fewest words possible. Every word, every character you add to a Gen AI prompt ups the energy usage. If you're searching just internet, you can add -AI and it won't use Gen AI at all. Learn how to prompt a little bit if you use these tools, be really specific. But you can put text in and ask it to create a reverse outline or find gaps in logic. Just know it's probably taking your information for use. So you want to be really careful with that. Notebook LM is an exception to Gen AI. It does not take up your text. It's free to use. It's really meant for college students. You can query PDFs, and you can also put in information and it will create an audio file. So it can make a podcast essentially for you if you prefer to get your information that way. And then I just learned about Ecosia. Instead of a Google or Bing search, they plant a tree for every search that you do. So really just use these things sparingly and carefully would be my ending thought there. And I'll share my slides.
Thank you, Alan, and thank you, Nancy, for this very interesting, very fun interactive session of how people do the editing. So with that, we conclude our workshop today. I hope everyone attending here, when you're preparing your manuscript, now you have a better idea what to do. At the end, we have a few announcements that we want people to be aware of. So as plant postdocs, we are putting up a database of potential plant peer review database and a potential plant speaker database. So the reason we do that, because when we talk with journal editors, we heard a lot that, okay, it's really hard to find reviewers. Like this paper, when trying to find a reviewer for a month, I reached out to 10 to 10 reviewers, they all declined or not respond. But what we heard from early career scientists, okay, I want to learn about review. I have time to review. I want to contribute my expertise in review. I can do very good, concise paper reviews. So there is some information missing between the two parties. So we want to put up this gap. So we have this database available on plantpostdocs.com. We're putting this link on the chat. So if you want to sign up for become a peer reviewer, please sign up this. We're going to share your information with journal editors, and they can decide whether to expand their reviewer pools. And also we have this database for plant speaker database. If you want to give talks, that is also something that that is also something we're interested in, and you can put into to get more opportunities. And with that, there's multiple ways to contact us. We have X, we have Blue Sky, and you can find the recording of this on our YouTube channel. And we have a Slack channel, and also we have a WeChat channel if you use WeChat. And we have many information on our website. And you can email us about any ideas. And please fill in the post-webinar survey. In the survey, there's also please tell us what you like and what you want us to improve and propose some new topics. And we ask you to put in your email if you want to receive future notifications about our events. We try not to spam people, so we want to be very careful. Okay, you really want to get us email, so please provide your email if you want to get future events notification like this. With that, we conclude our webinar today. Thanks for all the panelists being here. If you have other and for all the attendees to attend our webinar today. We didn't have enough time to get to the Q&A. All the attendees are free to go if other things to carry on, but you have something to discuss. Feel free to hang out a bit longer for some Q&A. Thank you.
Awesome.